Friends, today I am going to explain the concept of three words which are very important both for botany and zoology students. To understand these words, you have to understand as well as you have to prepare for the examination intention also. The words are biotechnology, genetic engineering, and recombinant DNA technology. As you are aware of all the three words, generally these three words are used interchangeably. वो इसलिए कि या तो उसके कॉन्सेप्ट्स के बारे में आप नहीं समझते या जो उनमें फाइन डिफरेंसेस हैं उनको आप नहीं पकड़ पाते. दो ऑल दिस थ्री वर्ड्स आर वेरी मच interrelated to each other, and as our information grew about the modern science, as you can see that as the word developed, in fact in 1970 biotechnology came into existence for the first time as far as word is concerned. This word was coined in 1917, though the information was existing much before that. Genetic engineering, this concept came in 1940. Recombinant DNA technology, this technology was developed in 1970 for the first time. We will take each word one after another, and from that information when you will get, then you can easily differentiate कि यदि इस पे क्वेश्चंस पूछे जाते हैं तो बायोटेक्नोलॉजी के क्वेश्चन में हमें क्या लिखना है, जेनेटिक इंजीनियरिंग के क्वेश्चन में हमें कैसे अटेम्प्ट करना होगा, और रिकॉम्बिनेंट डीएनए टेक्नोलॉजी को हाउ वी कैन एक्सप्लेन. So first, biotechnology. Biotechnology, as the name indicates, it is composed of two words, biology and technology. By using the bio from biology and technology, the word biotechnology was formed. From this derivation, you can easily understand that some property of biological organisms, whether the organism as a whole or some of its property, may be utilized by the techniques available to human beings and to produce certain new product. That branch of science is termed as biotechnology. When you will try to define the word biotechnology, then according to the definition of United Nations, any technological application that uses biological system, living organisms or derivatives thereof to make or modify products specific use, means वो कोई भी टेक्निक जो कि बायोलॉजिकल ऑर्गेनिज्म एज ए होल या उसके किसी स्पेशल प्रॉपर्टी को, जैसे हम एंजाइम्स को ले लें या किसी जीन को ले लें या किसी भी उसके किसी प्रॉपर्टी को यूटिलाइज करते हुए यदि हम कोई नया प्रोडक्ट ज़्यादा एफिशिएंटली प्रोड्यूस करने में सक्षम हो जाए, दैट टेक्निक इज टर्म्ड एज बायोटेक्नोलॉजी. फॉर एग्जांपल, टेक द preparation of curd. Here we use a bacterial culture. You take the preparation of bread. Here we use yeast. Then for fermentation purposes, for wine preparation, we need certain bacteria to ferment. So any of these products, when it is used by any of the property or an organism as a whole to produce or modify a product is termed as biotechnology. या इसी को हम दूसरे तरीके से भी डिफाइन कर सकते हैं, application of scientific and engineering principles to the processing of material by biological agents to provide goods and services. Means कोई या तो सर्विस हमें देना हो सर्विस मीन्स किसी कोई टेक्निक ऐसी हो जिससे कि हम ह्यूमन काइंड को कुछ बेनिफिट पहुंचा सकें, या कोई गुड्स प्रोड्यूस करना हो, जैसे कि बहुत सारे ड्रग्स हैं, हार्मोन्स हैं, एंटीबॉडीज हैं which can be utilized these days, particularly they are and become the common words for the treatment of COVID-19. For example, here you can say insulin hormone. Diabetes के patients के लिए बहुत important है. Today we are producing an artificial insulin hormone, synthetic insulin hormone, very much similar to the original human insulin. Then growth hormones, diagnostic tests for various diseases, all these things can be produced by biotechnology. Now we will try to go in certain history of this biotechnology so that we can understand the term in a better way. As I told you, this term was given by Karl Erke, a Hungarian engineer, in 1917, and he utilized to produce a better production of pigs by feeding them with certain plant products. That from that he deduced this term biotechnology. Now biotechnology has been around us since human began manipulating our environment to improve the food supply, housing and health. For example, making wine, beer, cheese and bread etc. बहुत सारे आइटम बहुत पहले से ह्यूमन बींग्स अपने यूज के लिए बना रहा है, या किसी न किसी बायोलॉजिकल प्रोडक्ट को वो यूज कर रहा है फॉर द बेनिफिट ऑफ कॉमन मैन. लेकिन फिर ये टर्म क्यों मॉडर्न टर्मिनोलॉजी की तरह यूज किया जा रहा है यदि ये इतनी पुरानी साइंस है? तो द रीजन बिहाइंड दिस इज दैट टुडे by the information which has come from the molecular biology and genetic engineering, now we can produce a large number of products. We can modify a large number of products. That's why the biotechnology word has become very common. Then the nature of biotechnology was again further changed by the development of recombinant DNA technology, because this technology has further improved the value of biotechnology, you can say. Now we have reached a condition that even various organisms can be used as a biological factories. Biological factories means as a production center. Only we have to provide the basic information, or we can provide the process and make the biological organisms to act accordingly. Microorganisms and eukaryotic cells could be used as biological factories for the production of insulin, interferon, growth hormone, viral antigens and variety of other proteins. Here I will like to give another term, that is union of recombinant DNA technology and biotechnology has now given again a new term, molecular biotechnology. Means recombinant DNA technology के जो इंफॉर्मेशन है उसकी वजह से अब बायोटेक्नोलॉजी वर्ड ही अपने आप में मॉडिफाई होकर के मॉलिक्यूलर बायोटेक्नोलॉजी के नाम से जाना जाता है. Now, another term, that is genetic engineering. This term was given by A. Jost in 1940. By this genetic engineering, we can now in a position to modify the genetic of any organism, whichever you want to change. When we go to its definition, then according to definition, genetic engineering is the act of modifying the genetic makeup of an organism. Means हमारे पास अब इतनी इंफॉर्मेशन हो चुकी है कि now we can change the genes of an organism according to our need and modify the organism as a whole. For example, you can easily understand by seeing this picture. Here you are seeing a normal rat. As you know that growth hormone is necessary for the total growth of our body. This controls कि हमारी साइज, हमारी बॉडी की साइज कितनी ग्रो करेगी, और निश्चित रूप से इस ग्रोथ हार्मोन के लिए some gene will be there. So if you determine that gene and transfer that gene into another rat which is responsible for the growth hormone, then we will develop a rat of much bigger size. Means by just changing the gene which is responsible for growth hormone production in a rat, we can increase the total size of the rat. Similarly, we can modify many other organisms according to our need, according to our requirement. Here you are seeing that fishes which are glowing in different colors. That is possible because of transfer of a gene which is responsible for fluorescent protein production in the body of the fish. Similarly, you can see that here the rabbit has been modified by using the green fluorescent protein. Its attractiveness you can easily understand. In the third picture, here the silk producing gene has been transferred in the goats, and now you can see the hairs that can be used as silk threads for the production of clothes or any other purpose wherever it can be used. Now the third term is recombinant DNA technology. Recombinant, as you know that this word recombinant, first of all you have to understand the meaning of this recombinant. Recombinant means new combination. If you understand this term, then you can understand this technology also. New combination, suppose हमारे पास 3 पैंट और 3 शर्ट है, और ये तीनों शर्ट अलग अलग कलर के हैं, तीनों पैंट अलग अलग कलर के हैं. अब यदि हम एक सिंगल कॉम्बिनेशन में पहने कि रेड शर्ट के साथ ब्लैक पैंट पहनूंगा, तब तो मेरे पास केवल 3 सेट होंगे. लेकिन यदि हम इन 6 कपड़ों को इंटरचेंजेबली यूज करना शुरू कर दें कि कभी ब्लैक पैंट के साथ रेड शर्ट पहने, कभी ब्लैक पैंट के साथ व्हाइट शर्ट पहने, कभी ब्लैक पैंट के साथ ग्रीन शर्ट पहने, तो और इसी तरीके से फिर दूसरे वाली पैंट जो कि आपके पास ब्लू हो सकती है उसके साथ फिर ऐसे कॉम्बिनेशन अपनाएं, तो देन जस्ट बाय चेंजिंग the shirt you can have many new combinations. Similarly, here we use these new combinations of DNA segments, different, you can say different genes in different combinations, and that technology is termed as recombinant DNA technology. Means here we use the DNA segments, are in plain words आप कह सकते हैं कि जीन्स को हम डिफरेंट कॉम्बिनेशंस में जब किसी ऑर्गेनिज्म में डालेंगे, देन द टोटल proteins which will be produced will be of different nature, and finally in the phenotypic expression of that protein will also be different. That technology is termed as recombinant DNA technology. The pioneers of this recombinant DNA technology were 2 scientists, Stanley and Cohen and Herbert Bayer. Both these scientists met in a conference and they discussed their areas of research. In 1972, Bayer was working on restriction enzymes. He was trying to understand how these restriction enzymes work, and Stanley and Cohen was working on plasmids. Plasmids, you know that they are the DNA, circular DNA segments present in the various, in fact, prokaryotes. Some plasmids are also found in eukaryotes, but mainly they are the elements of prokaryotic, particularly in bacteria. Both the areas were different. Cohen was very much astonished, as you also know, that antibiotics कोई डिजीज डेवलप होने के बाद एंटीबायोटिक्स कुछ समय तक तो काम करते हैं, लेकिन यदि वो लंबे समय तक डिजीज सस्टेन करेगी या आपने दवा ठीक से नहीं ली है तो आप पाएंगे कि कुछ समय बाद डॉक्टर कहता है कि अब ये बीमारी आपको इस एंटीबायोटिक के अगेंस्ट में रेजिस्टेंट हो गई है। तो ये रेजिस्टेंस कैसे डेवलप होता है उसके बारे में कोहेन स्टडी करना चाहते थे, एंड बोथ वर डिस्कसिंग अबाउट देयर प्रॉब्लम and finally they decided to work together. Then they isolated the different fragments of the DNA from animals and bacteria. वो ये रिस्ट्रिक्शन एंजाइम से कट करके उन्होंने डीएनए के फ्रैगमेंट्स को सेपरेट किया बैक्टीरिया और दूसरे एनिमल्स के द्वारा, और उसके बाद लाइगेट कर दिया, मीन्स डिफरेंट फ्रैगमेंट्स को जो डिफरेंट एनिमल्स और बैक्टीरिया से आ रहे थे उनको जोड़ा, उन्होंने लाइगेट कर दिया, और उससे जो प्लाज्मिड तैयार हुआ मीन्स द जीन इंसर्टेड फ्रॉम सर्टेन अदर एनिमल इनसाइड द बैक्टीरियल प्लाज्मिड, एंड दैट plasmid was again introduced into the E. coli. Then it was found that the normal genes of plasmid were expressed, plus also certain new genes were also expressed. This recombinant DNA technology was developed, and you can say that this was the first recombinant DNA molecule which was formed in 1972. Now, how we can define this recombinant DNA technology? When a gene of one species is artificially transferred to another living organism, it is called recombinant DNA technology. Means जब किसी जीन को एक ऑर्गेनिज्म से दूसरे ऑर्गेनिज्म में ट्रांसफर कर दिया जाता है और फाइनली वो कॉम्बिनेशन सस्टेन करने लगता है, देन दैट टेक्नोलॉजी इज टर्म्ड एज रिकॉम्बिनेंट। जिस प्रोसेस के द्वारा ये ट्रांसफर किया गया, उन्हें पहले सेपरेट किया गया, फिर जोड़ा गया और फाइनली एक किसी बैक्टीरिया में या दूसरे सेल में ट्रांसफर किया गया, देन दैट टेक्नोलॉजी इज टर्म्ड एज रिकॉम्बिनेंट डीएनए टेक्नोलॉजी। दूसरी डेफिनेशन भी है इसकी, प्रोडक्शन ऑफ ए यूनिक डीएनए मॉलिक्यूल बाय joining together two or more DNA fragments which are not normally associated with each other। मतलब कि जब दो different DNA segments जो कि normally नेचर में नेचुरल ऑर्गेनिज्म में नहीं रहते हैं, उन दोनों DNA segments को एक जगह ले आया जाए और ज्वाइन कर दिया जाए और फिर किसी सेल में ट्रांसफर कर दिया जाए, इंट्रोड्यूस कर दिया जाए, that technology is termed as recombinant DNA technology। We will try to understand that how this whole recombinant DNA technology process is done. Suppose this is a human cell, this is the nucleus, inside the nucleus we have the genetic material, some specific gene is taken out from this genetic material, here it is the human growth hormone. This technique is very complex, we will take it in certain another video, but first of all we can summarize the whole technology, and this is done with the help of an enzyme called EcoR1. This is the growth hormone gene with certain specific sticky ends. Similarly, this is the bacterial cell. Inside the bacteria there is one main chromosome and another DNA element is present. This is called plasmid. This is not very important for the bacteria, but it has play role in developing certain resistant properties. Now this plasmid is cut open again by the same enzyme EcoR1 which was utilized in case of human beings. The same enzyme is used to cutting this circular plasmid and it is cut open. Now when both these growth hormone gene and open plasmid is mixed, then they will join with each other, they will ligate with each other. Now in this bacterial plasmid, growth hormone gene has been inserted. This is termed as recombinant DNA molecule. Now this recombinant DNA molecule is again transferred inside the bacterial cell. This whole technology is termed as recombinant DNA technology. We will deal in great detail in our next video. But first of all, here it is taken to explain the word recombinant DNA technology. Again, here it is also the same experiment. Some gene, whole DNA has been taken out, and some part of the human genome is taken out with the help of certain restriction enzyme. This is the bacterial plasmid. Now both these are linearized and joined together to form a new DNA molecule, which is called recombinant DNA molecule, or it is also termed as DNA construct. Now this DNA construct is introduced into the bacterial cell, and this bacteria, inside this bacterial cell, the growth hormone gene expresses itself by producing new proteins which is normally not produced in a bacterial cell. This whole technique is termed as recombinant DNA technology. Okay, now finally all these three words has been replaced by molecular biotechnology. Molecular biotechnology, because now this knowledge has reached at the molecular level. You know that we have different fields of science. For example, molecular biology, microbiology, biochemistry, immunology, genetics, chemical engineering, cell biology. From all these fields, we are continuously getting new and new information. And all those informations is utilized for benefit or for improvement in our crops, drug manufacturing, vaccine preparation, diagnostics, livestock. Livestock means the animals which we rear for our benefit. So this technique which utilizes all these information, which is coming from various branches of molecular biology, microbiology, biochemistry, immunology, is used by a new field called molecular biotechnology for the improvement in crops, drugs, vaccine, diagnostics, and livestock. So friends, this is the explanation of these three terms. I think that now you are very much clear about these words, and if you find this information useful, then I will request you to please like, subscribe and share the YouTube channel, and wait for my next video to explain all these things further. Okay, thank you.